So we now have a much greater understanding of the molecular landscape of acute myeloid leukemia, and that's really come down to our ability to sequence genes and mutations at great depth. Acute myeloid leukemia was the first genome to be sequenced back in 2008, and it's probably the most sequenced tumor of all. Now that's taught us a number of things that we already knew. We knew it was a very heterogeneous disease, we knew that from its clinical behavior, and we knew that from the molecular test that we were able to perform before next generation sequencing. But I think what this has told us is that understanding was very much an underestimate. So we now have been able to sequence large numbers of AML genomes, and there are a number of collaborative measures to bring those all together. But there was a very large sequencing study recently from the Sanger Center in Cambridge and from the German AML study group sequencing 1,500 AML cases. And perhaps the most staggering thing from that was that there was more than 1,000 separate tumor types just on the basis of the number of mutations that they saw. And that gives us an idea of how difficult it is, because in effect, every AML is almost completely different. It is almost unique. However, they were able to collapse that down into a number of different groupings, 11 of those, and they conveyed similar messages in terms of prognostication. Some of them were lent themselves towards other therapies, and it allows us to develop a framework for personalizing the care in acute myeloid leukemia, and that's very exciting. So acute myeloid leukemia is a pretty rare disease, 4 in 100,000, and it's obvious that it's very complex, so 1,500 patients is not going to be enough. So there are a number of further collaborations where we're going to increase that to the tens of thousands, or certainly the thousands, and we'll be able to look at combinations of genes and what they mean for individual patients. We'll be able to compare individual gene mutations with novel and upcoming therapies and try and select the right patient population for the right drug and move us forward in our goal to personalize AML therapy, to select out those patients who need novel therapies, to select out those patients who could do without toxic therapies such as bone marrow transplantation, and to improve the outcomes in this fairly dismal disease.